I often have this love-hate relationship with schistosomiasis because it's such a fascinating disease in terms of its biology, but it's such a dreadful disease, and we need to have better ways to prevent it. So I'm based in the Natural History Museum, and our research remit is really about understanding the biology of the parasites, but also using that knowledge to actually develop implementation and to advance elimination in many of the areas. So just to point out, so we just introduced schistosomiasis here, but it's part of a group of diseases known as neglected tropical diseases. And we call them neglected tropical diseases because they're really diseases associated with neglected people. So the diseases of poverty, and other in terms of people don't have the infrastructure to actually prevent some of these diseases. So as you can see from this slide, there's various groups of these parasites, from protozoa, viruses, bacteria to helminths. A lot of these diseases are spread through vectors, particularly insect vectors, but there's also schistosomiasis, which is spread by snails, and snails are actually fundamental for the transmission of schistosomiasis. The burden of neglected tropical disease is very, very high. From this map, you can see that 1 billion people have one or more NTDs, and you can see where these diseases are globally. You can see they're very much widespread across Africa, South America, and areas of Asia. And in some of these places, people have up to 6 NTDs, so there is a really high burden of these diseases. So now focusing on schistosomiasis. So this is the disease that is my expertise. I've been studying this disease for about 25 years now, and there's still so many knowledge gaps around the biology of the parasites and understanding how best to prevent transmission, infection, and how to move towards elimination. As I said, it's very much a disease associated with poverty, lack of sanitation, and unsafe water sources. And we know that about 260 million people are infected or at risk. If we look at those pictures of the morbidity, the swollen abdomen, the blood in urine, and also that animal that is emasticated, the worms lay eggs, which causes that morbidity. You get large livers, large spleens, you get anemia through blood loss through urine, and then in animal schistosomiasis, you get chronic debilitation, wasting, and that causes huge economic burden as well. It's a very preventable and treatable disease as well, but it has a really complex life cycle. I often have this love-hate relationship with schistosomiasis because it's such a fascinating disease in terms of its biology, but it's such a dreadful disease, and we need to have better ways to prevent it. So let's go through the life cycle. So it's really fascinating. First, we're going to start right at the top of the life cycle, looking at the parasitic worms themselves. So around the blood vessels, in the blood vessels, around the kidney and the intestinal system, the parasitic worms lodge themselves, and they can live in those blood vessels for many, many years. And we know that people have remained infected for up to 30 years, so they're very long-lived parasitic worms. You can see in that parasitic worm there, they have, so we actually have them here in, I've brought them, they're preserved, they're not live, so please don't worry about them. They're about 5 millimeters in length. They actually have separate males and females, which come together to form a pairing mate. And their whole purpose is to produce eggs. The female, dependent on its male, and it'll pump out eggs, thousands of eggs a day, and those eggs then cause the problems associated with schistosomiasis. They become lodged in the tissues, they pass through the intestine and the bladder wall, and that causes that blood loss and anemia and all the other chronic symptoms as well. Okay, so they're small, but they're not very nice, okay? You can see in these videos, the worms up there is what we've dissected out to put in a petri dish. They're very motile worms, they're very active. And then these worms lay eggs, which will come out as people go to the toilet. They come out in feces or urine, and if they reach a fresh water body, those eggs will then hatch. And this video down on the shows an egg hatching and a larval stage hatching from that and swimming. The egg hatches through osmosis, water goes in, and then it bursts the egg. That Myristidia will live for about 24 hours. It seeks out a snail, and it will, if it finds the correct snail, it's not every snail, the correct snail, it'll go into that snail, and then over about 4 weeks, it will replicate itself clonally, produce thousands of larval stages. And this is one of the drivers in transmission. One larval stage goes into the snail, and thousands to millions come out. Comes out every day over many, many days, and up to about 3 months as well. So you can see that driving in transmission. That is one of the features of the disease that is so hard to control. So then it goes into the snail, and we have snails here. We have live snails. We culture snails in our laboratories at the Naturalist Museum. This is one type of snail. It's called Balinus truncatus. It's very widespread across Africa. It transmits the form of the parasite that goes into the bladder system. They are small. They're hard to find. It's hard to find them when we go and do field work. So as I said, once in the snail, it replicates. It comes out in the final larval stage. This is called Sakari. It actually has a head. It actually lives for 24 hours again. It has no feeding source, so once it's used up its energy, it actually dies. The head will stick to the skin of the mammalian host that it wants to infect. It releases an enzyme. It allows it to go through the skin. You don't feel it. It's microscopic. And then it goes into the blood system. The juvenile larval form then goes through metamorphosis, and it gradually develops into the adult worms. So you can see how complex the life cycle is, but how well adapted it is, and how those different life cycle stages change. And we still know, we have still fundamental knowledge gaps about those different life cycle stages as well. Once inside the blood system and it reaches those adult worm stages, it goes into the intestinal system, the worms pair again, and then it releases lots of eggs, which goes on to cause that damage to the tissues. And that build-up of chronic morbidity. Okay, so drivers in transmission. I said snails really drive transmission. Water contact drives transmission, and that contamination, lack of sanitation and behaviours as well. These are very classic environments that we see schistosomiasis. This is where we've done a lot of work on the ground. We have women using water bodies for washing pots, daily activities. We have a lot of children playing in these water bodies, and we also have water bodies that are used for agriculture. And as I said, it's associated with sanitation, behaviour. We have open defecation, we have open urination. And these people infected are churning out thousands of eggs each day, which go into these water bodies, and you can see that's really hard to control. There is a treatment. Praziquantel is the only drug available for the treatment of schistosomiasis. We have no alternative, and this is one of the worrying aspects of control, and this is what the project I'll talk about is focused on. We can put in safe water sources and sanitation. We can do snail control as well. That's often not very environmentally friendly, and it's also very difficult in very big water sources. And we can implement behavioural change, but again, changing people's behaviour is also very difficult unless you provide alternatives. So let's look at those snail habitats again. We've seen the smaller snails. They are really hard to find and to actually map them and see where high-risk areas are. And then the other form of the snail, which we'll have a look at now, is the Biomphilaria snail, very widespread in Lake Victoria and also many other water bodies across Africa. These again transmit the intestinal form and are at very high numbers in these water bodies. They're a bit easy to find, but they are also very, very widespread and very, very difficult to control. Okay. I just wanted to put this image up because this really highlights the amount of larval stages that come out of individuals. So this is the urinary form of the parasite. This is what I've done here. This is work in Zanzibar. We filtered out the eggs from a single child in Zanzibar. Zanzibar isn't meant to be a place where we have control of schistosomiasis, but we have small pockets of infection. This child had thousands of eggs coming out and thousands of larvae. So you can see that even with one child that's missed treatment or it has a high infection, this can really lead to problems with control as well. Okay, so on to the project that we've got funding from the Wellcome Trust. So this is really to look at the drug itself, praziquantel. As I said, we have one drug to control schistosomiasis, and we're very worried about low efficacy, a low performance of the drug, but also potential resistance or the evolution of resistance. So the project is called RESIST, the Resistance Evaluation and Surveillance Initiative for Schistosomiasis Treatment. So we're working in Zanzibar. We've been working in Zanzibar for about 30 years now, but before my time, my mentors were working there before I took on some of this work. doing snail surveys, doing treatment and implementation of prevention strategies. And Zanzibar is 2 islands off the coast of Tanzania. And if we think we can eliminate schistosomiasis, this is probably the best chance we've got because it's an island population. We have 1 species of snail, which is involved in transmission. We have 1 species of the parasite as well. But treatment, so this gives you some data from what's called the Zanzibar Elimination of Transmission study, which was funded by the Gates Foundation between 2012 and 2017. And within this study, they were implementing treatment biannually, so at a very high dose, doing behavioural change, snail control. But what we actually found is we had persistent transmission in some places and no long-term prevention by even doing that implementation. This graph just shows you the ups and downs of prevalence in different communities. And you can see in red there, even at the end of in 2021, where we've had long-term drug treatment, we still have areas of high transmission. But it can be very, very dynamic as well. It's very much affected by environment, climate, and lack of, or when interventions stop as well. So this just shows you how those hotspots of transmission change over time as well. It's just a very nice visual aspect. And we were looking back at the data of 2017 to identify we have some areas which are really hard to actually gain control of schistosomiasis. And we have huge bounce back of infection once we stop intervention. So the overall aim of RESIST is to understand whether praziquantel resistance has potentially contributed or is contributing to the existence of persistent schistosomiasis on Pemba, the Northern Island of Zanzibar. And if we find it, how do we address this going forward? It's very much a collaborative project. It is very much led from the ground by Saeed Mohammed, who runs the labs in Pemba in PHL. Swiss TPH support them in the fieldwork epidemiology. My team at the Naturalist Museum, we collect the parasites and we're doing a lot of the genetics. And then we have a modelling component. Erasmus University are leading the modelling of transmission and trying to understand or predict better ways of all the treatment impacts that are going to happen. So very briefly, I'll show you the work plan, and then I'll show you some of the data that we've got. We've only done the first year. We've done a lot of the field work. It's been very, very busy, but we've also learned a huge amount as well. So the work plan starts with surveillance, and the way we're doing surveillance is we're filtering urine, we're looking for eggs, and we want to gain an understanding of the true prevalence levels. We're working in 17 places in Zanzibar, 17 schools in fact, and within those schools we have participation from 800 to 1,000 children within each school. So it's a large number of participants. This is a very big study. For 2 of those schools, these are 2 hotspot schools that have always been hotspots for as long as it's been recorded. We have high levels of infection. It's very difficult to get down to low levels, and when treatment is stopped or interventions are stopped, we get bounce back very, very fast. In those 2 schools, we're doing intensive surveillance. We collect urines over 5 days to try and get a true prevalence level. And in the other 15 schools, we survey children at one time point to get a baseline prevalence for those low-level areas. We treat everybody then. We do mass drug administration, not just those infected, but the whole school community, even if we know they're not infected. We wait 2 weeks, and then in those 2 intensive schools, we're collecting urines again after treatment. We're doing over 5 days to see if anybody is still infected and if they've not been cured from their infections. The data we're gathering is about spatial infection levels and intensity to build those models, but also to give us a better understanding of what's actually happening on the ground. Large numbers of treatments will be given to actually support elimination and to prevent that chronic morbidity build-up in these communities. And we're very interested in those treatment outcomes, looking at cure and egg reduction rates. And then from my side, we're very interested in the genetics of the parasites as well. And this has really come about because of some groundbreaking work in 2019 by 2 groups from the USA, which have discovered how the drug praziquantel actually works on the parasites. Even though we've been using these drugs for decades, we did not know how it works. And this work has actually shown that it's targeting a certain area of the parasite called the TRPM channel, And now we can look at mutations in that channel to actually track how well the drug is working. And that's very much what my team in the museum is doing from these collections. This work is very difficult. It's logistically it needs a lot of resources, it needs a lot of teams, but it's driven very much from the ground. There's a lot of community engagement, working with the communities, the schools, the teachers, and the local ministries of health. We work with them to develop the ethics, we register all the participants, we get the consent from those children, and very much work with those individuals to actually allow this study to go forward. The infection levels are screened through diagnostics. They're looking for eggs in urine. Again, this is a logistically challenging operation, looking at 17,000 plus urines. The team on the ground in the labs in Pemba are excellent. They are very well, they've done this through many treatment programs. The way we do this, the urine is put through a filter, the eggs are captured in the filter, and then the microscope is used to actually look for those eggs. And then we can get an intensity level and a prevalence level from those communities. And then treatment happens. This is one of the most rewarding parts of the work that I've been doing on Zanzibar, being involved in these treatment, the administration of the treatment. So the way this is done as well, we go into the schools. The children are very used to being given treatment, and the teachers really support that. Children come into the schools, we go in with the team to actually administer that treatment. The children actually get fed at the same time. This helps the metabolism of the drug, but it also allows them to have a meal during their school time. So it's often an incentive to come in to get that treatment as well. You can see the children actually participating in this, brewing up the porridge in the morning to actually give the children before they're actually treated with the drug. And then they're given the treatment. It's based on their height, and the height determines how many tablets they have. And then they're given the treatment. They are observed taking the treatment, and they're observed to see if there's any side effects, and they're observed to see if they're vomiting or hiding those drugs as well. We do get children that hide the drugs because they're actually not very nice to take. So we're trying to get really good coverage and observe treatment to actually make sure this treatment is being taken at the appropriate level and to have the maximum effect. So we hope to reduce treatment, reduce prevalence, prevent that chronic morbidity, improve the overall health and well-being, and eventually hope to move towards elimination. And so on the genetic side, how do we do this? So it's actually not that difficult to collect the parasites. As I said, this is an image of larvae coming out of a single child. We capture the eggs, you put them in water, they hatch very, very quickly. Within minutes, they're hatching. And we capture single larvae using a laboratory pipette, and we store them on these DNA storage cards, and we can capture up to 60 larvae per child, and we store them on these cards, and then we do the genetics on that. So what we're doing is we're going to look at about 576 myricidia from these different communities, particularly those post-treatment, to see if we've got any mutations that are appearing that show we've got lack of efficacy of this drug as well. Okay, just to finish off, very preliminary data. One of the key things we've found so far is that we have much higher prevalence than we thought, particularly in these hotspot schools. We know that if we sample a child on just one day, our prevalence is about 20%, but if we look at that child across 5 days, our prevalence jumps to about 35%. So we know that our routine diagnostics are not good enough to giving us true prevalence levels, and we have much higher levels of infection than we thought. Cure rates are about 76%. In our mind, that is not good enough. We need it much higher. And again, we're very much looking at these post-treatment parasite populations to see what's actually going on there. And our next steps is to analyze the data from all 17 sites, look at the genetics, and then also advance the strategies to enable schistosomiasis to move towards elimination in Zanzibar, and that's our ultimate goal. And then in the broader scale, what is this project contributing? Well, it'll really help us to know if we need an alternative to treatment or better interventions. We hope this kind of surveillance approach is going to be adopted across Africa, because we need to look at these parasite populations across all endemic sites. And we also want to advance WHO guidelines for schistosomiasis control and elimination, and also in terms of diagnosis to give us true prevalence and intensity levels as well. So there's much broader aspects that we're considering. And with that, I'll stop there. Lots of thanks, particularly the Zanzibar team. This image down here is the team of all the collaborators when we were working in Zanzibar, and all the participants as well that take part in this study. Thank you. Yes, snail control is needed, but we need a novel way of doing it. And also, we use Zanzibar as an example for snail control sometimes, but we can't do that in Lake Victoria. Lake Victoria, it would have no impact at all. So we need to think about how we're broadening that approach as well. So behavioural change did have an impact. What we were doing with behavioural change, we were giving particularly women washing platforms, so they were not going into the water bodies. And those behavioural changes were very much about culture as well. Women like to go and wash their clothes together as a community. It's a way of coming together and communicating. So we provided those facilities for them, and that actually reduced transmission, and also they were not taking their children to the water bodies as well. So it did have an impact. It's the sustainability. When we go back now and we see those platforms that we've built, they've degraded, the women are not using them anymore. We put in latrines as well, pit latrines, and again, they were not maintained, so they're not being used again. So it's sustainability. Which is hard. I think in terms of vaccines, absolutely. If we can come up with a vaccine that is going to be good enough, I think that is a way forward. At the moment, I think some of the data shows that it's going to be in combination with treatment as well. It's vaccine development for helminths is hard. You probably know more than I do about that. But it is the ultimate intervention in the long run, but also is infrastructure and putting those alternative interventions in as well. Yes, absolutely. So children are a lot of the mass drug administration is done in schools because they are a contained unit. So you can do cohort studies with children. They're also the ones that are most infective and the ones that are contributing to transmission at the highest level. If we had all the resources available, then we'd do community-wide participation as well. Yes, so in terms of climate change, there is two factors to this. So we know we have schistosomiasis, or they've just announced actually they don't think it in Corsica, which is the Mediterranean island. Is the urogenital form, so it was the transmission through urine that actually occurred in Corsica. There is data suggests that it has it's enabled itself to establish there because of increases in temperature. The snails have always been there, so it was just a factor of people migrating into Corsica and introducing the parasite, but that slight increase in temperature is probably allowing that transmission to continue. So with climate change and introduction of schistosomiasis into new areas, you have to have the three factors. You have to have water contact, you have to have excretion of eggs in urine or feces, so bad sanitation, and you have to have the snails. So in some areas where you've got the snails, but you haven't got people in the water or bad sanitation, you're not going to have schistosomiasis. So again, you have to have those three factors in play, which is important. In terms of climate change, so again, we know that we're getting floods. That obviously in places like Africa is going to introduce schistosomiasis into new areas. We also have droughts, so it's coming out of certain areas as well. We also know that too high temperatures, the snails are not happy, and also the parasites are not happy. So as temperatures get to a really high level, that's also going to be detrimental to schistosomiasis. So there's a lot of things to think about in terms of climate change, and not to just always go with the, yes, it's going to have a massive impact as well. So. So snails are very attracted by food. So that is their main attractant, and there has been groups that have developed, it's like a snail gel, and actually we feed our snails on it, and as soon as you put it in, they are on it. And there is a group actually wanting to trial this in sites to actually attract snails to a certain area so that they can be removed to actually bring down the snail population. So yeah, so that would be a nice way to have. And snail traps, they would love to develop snail traps for that. So
thank you so much. I think if everyone can join me again in saying thank you to Bonnie and for a really interesting talk.